uh, Dr. Spitali, who spoke earlier and threatened to speak at you again. He is now going to talk about this.
Okay. I promised that I'd do a better job with the microphone, so I'm going to do my best. It's me again, unfortunately for you. So I want to talk a little bit about just how hard it will be to actually serve the patient community from a therapeutics development perspective. The diseases that we're interested in treating at Savannah will require the production and scale of a very hard to make drug product. And one of the things that I very strongly believe is that for quite some time, when I say we, I mean a field, have been trying to treat devastating CNS and other genetic diseases with therapies that don't really scale to the number of patients. And that has shown itself in a few ways. One is that a lot of the focus on innovation and drug development is primarily to serve very rare diseases, which are, of course, very important. I'm not saying that. But they don't come with solutions where you can treat patients that have larger patient populations, very disparate populations in not only their biology and their disease onset and progression, but it's really a scale problem. When we think about traditional modalities in here, we have gene therapies, which have made a lot of progress scientifically, but unfortunately are still not really where we need them to be in treating CNS diseases at scale with a great safety profile. The other one is in bone marrow transplant and doing genetic modification of hematopoietic stem cells before they're reintroduced in the body. And then we saw earlier from Dr. Kufal's work that could have some potential issues in the clinic. But the more important thing is that for these therapies to have success, to make the transition from an academic hypothesis to a therapeutic hypothesis to a small clinical trial to then approval to then be a commercial success, we need to make a lot of development in our innovation and the way that we develop those therapies. $2 million a patient to get a curative medicine is unacceptable, frankly. And we need to be able to develop technological advancements that allow us to merge many different disciplines of emergent research to come up with new ways of treating them. That is part of our mission at Savannah, is to think a little bit more outside the box in how we develop these therapies to try to overcome those commercial and scalable limitations that I mentioned. So I want to talk a little bit about where we are when thinking about microglia cell therapies. Dr. Shetarevian did a good job of introducing the concept of induced pluripotent stem cell-derived microglia, so I want to talk a little bit about that and the implications of that when making these kinds of medicines going forward, and also some of the other things like being able to control microglia cells or microglia replacement, and what does it mean to think about it from now to a commercial success, which is what we're ultimately after to actually make these things work. There are several examples where bone marrow transplant or HSCT transplant or other advanced cell therapy transplants have made technological bellwethers. The ability to manufacture, create a drug product under CMC and release that has been met, but fallen down commercially because of the lack of scalability. And that really, I think, is a detriment, of course, to patients, because when we think about making drug products, we want those things that are really going to translate. So I want to talk a little bit about that. So here's a quote that I'll start with. Microglia replacement can mature from early breakthroughs into a generalizable platform across neurological diseases. That makes sense if you think about them as a central conduit to many different types of diseases. And it makes sense if you think about them as a node in a network inside the brain. So what do I mean by that? Apologies again, some of the colors are not going through here, but you could kind of abstractly think about the brain as this network of cells that are interacting with each other all the time. There are different nodes that are constantly signaling to each other and talking to each other. And inside the central nervous system, you could have a population of disease microglia. It may be beneficial to add healthy microglia into that system to tip that system from a dominated disease population of microglia cells to one that has healthy microglia cells that are co-inhabiting with those disease microglia, and they can compensate for faulty microglia function. For example, in ALSP, as I mentioned before, it's a little bit more linear. There's a loss of number of microglia cells. Introducing healthy microglia cells into that system can compensate for lost cells. It is also true that you can have a full population of disease microglia that are not supplying the brain with the right enzyme, or not supplying the brain with the right signaling peptide, or something's happening where the microglia function is not quite working well. And you can introduce healthy microglia into that system, and those healthy microglia cells will resupply the brain with a lost protein, a lost enzyme, or a lost signaling molecule at the amount that would be required to compensate for the lack of that function by the disease microglia. We call that a stoichiometric amount, which is just a fancy word for there's enough molecules around to do the function. There's also a scenario that was also brought up in Dr. Shadarevian's talk where we could think about the concept of targeting microglia specifically with microglia depletion agents to remove diseased microglia and enable healthy microglia that are introduced into the system to populate that open space where those diseased microglia are now gone because we've selectively removed them. Now, a lot of this stuff may actually seem like science fiction, like you're going to introduce cells into this system, and what does it mean? What's the potential impact for that? But there have been several preclinical animal models that are routinely used to develop other modalities, modalities defined here as gene therapies, small molecules, antibodies, et cetera, where those animal models are used to test the ability of those drugs to have efficacy on those models, where microglia transplantation or microglia replacement has demonstrated curative potential or disease modifying effect potential. So what we're thinking about here is a general way to introduce healthy microglia cells into the system and have it be done in a generalizable way to think about correcting faulty microglia dysfunction or microglia biology. So the implications are quite large here. So to think about the consequence of this, there are three major challenges that we as an organization are focused on doing. Our goal here is to translate the amazing research and development that's been done by our colleagues and us that have helped form the company and try to find a way to translate that into products. We have to be able to manufacture these cells at scale. We have to get these cells somehow inside the brain or inside the full central nervous system. Remember, I mentioned that these cells are in the ears, the eyes, the spine, and the brain. And it would be transformational for us to be able to monitor and measure cells where they are when we transplant them. Dr. Shadarevian brought up the fact that we've learned a lot as a field from the CAR T cell therapy space. These are T cells that are introduced into patients into that system where those T cells are programmed to go after cancer cells and destroy them. Now, the beauty of that approach is that you can monitor the blood. You can introduce them into the blood, then take a blood sample every few months, and you can monitor where those cells are in that blood. In the brain, it's much more harder to monitor where the cells are and to monitor what they're doing. So to have tremendous breakthroughs in biomarkers or imaging agents that would allow us to monitor microglia cells would transform the field, okay? So the first thing that everybody should appreciate, I think, is that making microglia cells at scale is really hard. It's a challenge to be able to produce them under CMC conditions where the drug product is characterized and standardized the same way that you make every single dose. And if you think about it, if you guys take Tylenol, the manufacturer of Tylenol has to release that pill under the same critical quality attributes, means that it has a particular chemical structure, that chemical structure is pure, that chemical structure is stable, that allows you to make that pill that we take, right? So cell therapies are much harder because they're living medicines, right? But they still have a set of critical quality attributes that tell us, is it the right cell? When we make those cells, are they pure? Meaning that you have your cells that you want and there are no other cells that you might have introduced because you manufactured them. And is that cell potent? Meaning that when it gets dosed into the patient, it's going to do the thing that it's designed to do. It's actually going to function and do that disease reversing functionality that Dr. Shedaravian talked about in his discussion. Not only that, but it's really hard to distribute these drug products. So they have to be frozen. There needs to be a really clear chain of custody when they comes out of the manufacturing suite to go all the way to the clinical site to then be dosed. There's a lot of different moving parts that have to be organized, right? And I bring this all up to say that these things are all doable. So I don't want anyone to think that they're not doable problems, but just for us to pause as a group, focus on ALSP, mostly because selfishly, I believe that ALSP is a disease that can be cured with cellular medicines. So we have to understand as a field, what are the things that lie ahead of us and our opportunities to define this together. The technological achievements of bone marrow transplant and hematopoietic stem cell transplantation have resulted in curative medicines for many different types of aggressive malignancies and cancers. There has been some thought of utilizing bone marrow transplant, or HSCT, for the utility of diseases like ALSP or other microgliopathies. For diseases where they are metabolic disorders, where there's a missing enzyme, or lysosomal storage diseases are a great example, giving back a small amount of cells that can go and pass the blood-brain barrier to supply a multi-turnover enzyme at high enough levels to digest a substrate of interest. Bone marrow transplant has actually demonstrated remarkable efficacy in patients. There is a little bit of a challenge here, though, because bone marrow transplant in general has a lot of issues in the clinic. If you speak with clinicians, the way that we precondition patients is not standardized across the field. The way that we understand the implications of the pre-treatment regimens on the integrity of the blood-brain barrier and also the stability or senescence of the cells that are present in the brain is not really well understood. And the ability to get those cells past the blood-brain barrier is something that is still under pretty heated debate, and I'll talk about that in a few moments as well. In our company, we think about this through a few different lenses. One is, what is the drug product doing? In this case, what is a stem cell-derived cell going to do that comes from a hematopoietic stem cell? And what is a stem cell-derived product going to do that comes from an induced pluripotent stem cell, which are very different cells. They both say stem cells, but they're very different, okay? So in an autologous gene-corrected cell, the cost of making and scaling autologous gene-corrected hematopoietic stem cells to larger indications is very prohibitive. The process of taking blood out of a patient, gene-correcting the genetic alteration, which is definitely what would be the case for ALSP, and then making those cells and then putting them back into patients can create major bottlenecks when it comes to translatability and commercializability in the market. And something that I really struggle with, you can ask Alana, I struggle with this, yeah, you can ask Alana, is it's hard to put a rosy picture on this because we want to be able to, as a community, rally around the best thing for patients. That's our number one goal always. But the fact of the matter is that there have been several curative medicines that have been developed around hematopoietic stem cell transplant treatments, curative medicines, that have been abandoned commercially because of the limitations on the commercializability and cost to payers for the sake of patients, okay? And I struggle to bring that up because I think it's a very difficult thing to talk about in a room full of patients and patient advocacy groups. And the only thing that I can say to you guys is that I think there is a desperate need to come up with innovative solutions that will enable commercializability at scale for the sake of patients and their patient advocacy groups. That is our mission, okay? So autologous gene-corrected cells, which will be necessary to perform whenever you are dealing with a genetic disease where there needs to be a genetic correction for those cells to be reintroduced to the patient for therapeutic modality. The cost is very high, they're hard to manufacture, and you create one dose per run, right? As we think about using these kind of microglia or microglia-like therapies into larger indication space, it's going to be really hard to break through because it creates a perfect storm for payers, for clinical sites, for infusion sites, and for manufacturing. All of those things need to be interconnected to make these drug products work. For allogeneic HSCT, you can get a donor cell from a healthy donor that doesn't need a gene correction, okay? The cost of that is lower. It's donor limited, and it's donor limited in scale. And one of the things that Dr. Kufal brought up in her presentation was that the best case scenario is to have something that's autologous because matching to patient has what's likely to be the best outcome for patients because it's your cell going back into you. Your cell is not responding to that. It's likely to have the best outcome, okay? There's another way to think about this, which is autologous gene-corrected iPSC-derived microglia. This is where you would take blood from a patient, reprogram that into an induced pluripotent stem cell, take that stem cell and genetically modify it to correct the genetic mutation, and then turn that stem cell into a microglia. Dr. Shedaravian showed you guys what I think is breakthrough preclinical efficacy to demonstrate the utility of that approach, sourcing blood from patients from Dr. Zolick's clinic, and then genetically editing those induced pluripotent stem cells, turning them into microglia, and then reintroducing them into the preclinical pharmacology model that he talked about. The issue with that is a very, very hard to scale approach. It's just as hard, maybe even harder than autologous gene-corrected. Speed of manufacture is months, and it's one dose per run. The long pole in that manufacturing is the gene correction. So being able to use CRISPR modification or viral modification to change the piece of DNA that's not what we want and turn it into a piece of DNA in those cells that is the healthy genetic sequence. That's the hard part there, okay? And then you have allogeneic healthy donor cells So those would be allogeneic induced pluripotent stem cells that are differentiated into microglia, and then you can make many doses of those cells at scale. And this comes again with some challenges for a lot of these gene-modified cellular approaches. You need to have immune suppression to help with the engraftment. You need to have other approaches. I think the central nervous system is probably the best case scenario in any cell transplant. It's not necessarily an immune privileged organ as it's been historically perspective, but an immune depleted organ in comparison to the rest of the body. So we and others, and with our co-founders lab, Matt Blurton-Jones's lab, we're starting to understand the ability to engineer allogeneic cells to be immune evasive, so not really responding to the donor or the recipient's immune system. And the point I want to make here is that of all these things, we think that the technological achievement is focused here, because that allows us to think about making products that can scale, can be off the shelf, meaning that we go into a freezer, we take a vial out, and we prepare it to be dosed. And these things can be bankable. So we believe in our company very strongly that the technological achievements deserve to be solved to make real products that can meet the demands of the marketplace and the patients at scale. That's our mission, okay? So in better scenario, this is a person, this is a cell going into their spine and up their spine into their brain. Just suspend your disbelief, but that's what's actually happening there, okay? So once we've made a drug product, how hard is it to get the cells into the brain tissue? There's a couple of barriers. You go from the blood to the CSF. Everybody okay if I close this? Sorry, thanks. There's the blood to the CSF, and then the CSF into the brain. So the blood-brain barrier blocks systemic cell delivery. As we were talking about a little bit earlier, there might be ways to get these cells and coerce them to go across from the blood into the brain. Brain injections require precise neurosurgery. Now, the one thing I'll say there is I'll pause on that for a second to say that stereotactic neurosurgery is something that's been around for a very long time. There's a lot of ways to think about this and to get that into conventional neurosurgical suites. There's also more sophisticated ways to introduce these cells into the cisterna magna or intracerebral ventricular, into the ventricles, to the cerebral spinal fluid, or into the lumbar spine, like we were talking about a little bit earlier when Dr. Zolik was asking about delivery. There's a lot of ways that we can think about this, okay? Cell delivery can reduce cell viability. Cells distribute poorly through brain tissue, and engraftment is limited by the host environment. So there's a lot of big challenges that you have to think about when getting these cells to go into the brain. The good news is that microglia cells are incredibly mobile. They're very proliferative, as we were talking about earlier. They are incredibly migratory. They can move all around the space. So we actually can leverage that biology to let the cells do a lot of the work for us because of that. Now, when we just think about introducing cells from the peripheral environment to get them into the brain, this is kind of where we're at with our kind of best case scenarios right now, okay? This is well recorded in the literature. So non-myeloablative chemotherapy, you get about 1.3% chimerism. Chimerism here is defined as the number of green cells to the number of orange cells. So 1.3% of all microglia cells in the brain would be green, the rest of them would be orange, okay? So you have this chimerism that's set there. With busulfan myeloablation, that can get up to almost 7%, a much more aggressive way, highly aggressive busulfan, you can do that. Treosulfan myeloablation, total body irradiation myeloablative, about 8%, and multiple myeloablative hematopoietic stem cell transplants can get to about 16.3%, but that's after multiple aggressive pretreatment regimens before dosing cells. So my point here is that the brain is a very large physical organ in space. When you're dealing with microglia biology, the bad part about it is that microglia cells are everywhere, right? And if the microglia cells are driving disease, it's really hard to think about replacing all of them and getting all of them out, or trying to get to a best case scenario, which is that every cell is a green cell, right? So that's the challenge that's technologically advancing for us. As Dr. Shetarabian pointed out, the Blorton-Jones lab has been the pioneer in a concept of using, and frankly, repurposing clinically approved drugs that target CSF1R on microglia. So the idea here is to target that gene with a small molecule that's specific for CSF1R. You then compromise the integrity of the microglia cells. Those cells become depleted, so you're removing the dysfunctional microglia out and replacing them with healthy microglia. So if you're fortunate enough to be able to treat with this microglia-specific drug, these cells will come out, and then you add in healthy cells that are engineered to be resistant to that drug, then you can replace the orange cells with the green cells, okay? So essentially what you're doing is, that's fine. A simple way to think about the brain is it's a one-car garage, okay? You want to get rid of your bad car, take the bad car out, put the good car in, okay? That's what we want to do with microglia. These are the reagents that we've used to do this in rodent models. Here's the dosing that we do in rodent models. Here's the human equivalent dose of this, and here's the human clinical dose that's been approved for these two drugs. These two drugs have not been clinically approved yet. There's no human data. But one of the points I want to make here is that it could be that the ability to translate the work that we've done in rodents into humans based on dosing regimens may actually be a possibility. So we're really excited to continue to lead at the forefront of using these molecules, but also developing new chemistry that is more specific to microglia, has a better safety profile, and can enable us to control the microglia cell population in patient brains to swap that transplantable niche. So our ultimate goal here is to produce these cells at scale and then be able to replace them, okay? You could think of it like a bone marrow transplant, where there's a conditioning regimen that happens to clean out the bone marrow compartment, and then new cells are introduced because of that. The exception here is we want to be much more targeted and specific to microglia without broadly used myeloablative or chemotherapeutic interventions that are traditionally used. The last big topic that the company is excited about is developing novel reagents to visualize where microglia are physically in the brain, how their populations change over time, and to be able to directly read out microglia populations or dynamics for the sake of clinicians and patients to better understand what these drug products are doing when they're administered into patients. So here's a couple of examples. This is a CSF1R inhibitor that has been functionalized with the C11 positron emission tomography emitter. And when this molecule is dosed into patients, you can see that it generates a heat map of microglia density. Where this molecule is binding is where microglia density is high, so the red and orange regions, and where microglia density is depleted is in the white and blue regions. And something that's quite fascinating to think about this is if you look at the overlay of this population density and where a lot of the MRI white matter lesions occur due to microglia depletion in ALSP patients, they overlay quite nicely, especially at the areas that are periventricular or in the corpus callosum. So what this means to us is that we can think about using these reagents to better understand microglia biology, population dynamics, and better inform clinicians and patients on what's going on inside their central nervous system. And here's another example where this is a TREM2 antibody that's functionalized with a positron emitter, and this is looking at the density of TREM2 just through a different brain section. So the point I want to make here is that there's a lot of precedence for thinking about this problem, moving in the right direction, and we're excited to continue to develop these kind of technologies alongside that. Okay, so Savannah's mission is to scale the manufacturing, enable the dosing, and establish novel clinical methods to track iPSC-derived microglia silent medicines. We're excited to apply this approach. Sorry, a lot of this stuff is not coming through. That's my fault. We're excited to apply this to ALSP. We think it's a great starting point to think about microglia cellular medicines, how to produce them at scale, how to get them to patients to where they need, and to be able to introduce them into the CNS for them to have transformational efficacy that's consistent with what my colleague discussed earlier. I'll just pause here to mention that we've made a lot of progress in probably what I think is the biggest lever to pull in stem cell derived products for manufacturing. When we first started this journey, it was going to cost us $1 million to make a GMP dose of microglia medicines in hundreds of millions of cells. Each person has about 5 to 7 billion microglia in their full CNS compartment. We want to be able to introduce these at similar scale to give them the best opportunity for drug product efficacy. We now can release a clinical dose for under $50,000 a dose. We will continue to drive that down by over 50% by the end of the year. We will be on target to release that for under $25,000 a dose. To put that in perspective, the average cell therapy costs anywhere from $300,000 to $500,000 a dose. So we're about 10x under that. We are very strongly committed to bringing solutions to patients that actually payers will pay for and patients will be able to utilize as part of treatment regimens. We're committed to making that major technological leap that's necessary to do that. We have also developed formulations are established to ship this drug product anywhere in the world. Comes out from central manufacturing. It's frozen in a little cryo vial. That cryo vial can be shipped to anywhere. We are very close to being able to say that we can receive those cryo vials at a clinical site. They can be thawed, put into a syringe, and that syringe can be taken to the clinical site and ready for dosing. It seems a little bit, well, yeah, that's obvious. Of course you want to do that. But all of these things that are important for chain of custody are really critical for us to be able to bring these things to patients. So we've made a lot of progress in that. And we also think that microglia cells are likely to be very important for many diseases, as we've been talking about. So it's been a recurring theme from today's session that I appreciate. Here's our executable path. So we, alongside our colleagues, published a couple papers in the journal Neuron, that some of the data that Dr. Shadaravian talked about. We've also published with always lovely and talented Dr. Zolik a couple of papers in movement disorders and analysis of clinical and translational neurology. Some clinical studies that we've done with his group. Thank you so much for the support on that. We also, with Dr. Zolik's help, completed a pre-IND meeting with the FDA earlier this year that went very well. It was very aligning and galvanizing for our organization, so we're thankful to the FDA for the support. And we are aiming to be in the clinic sometime in 2027 and organizing ourselves. We'll be working with Dr. Zolik on that effort as well. So that's it. I will pause for any questions. Thank you for your time. You.
We have time for one question.
Just one.
So
it's one question, but has 3 parts.
okay, very simple.
Okay.
All the patients who died after the standard bone marrow transplantation died in the first month or so.
Yeah.
Due to myoablation.
Yeah.
Now, you are in your disease. You are dealing with dysfunctional microglia.
Yeah.
Probably sick microglia or already gone.
Yes.
Why you need myoablation in your future trial? Maybe
I don't.
Maybe you don't need that.
I don't. No, I don't need myoablation. So we will not use myoablation in our trial. Thank you for bringing that up. We will not use myoablation in our trial. We do not need to pre-treat patients with myoablative procedures or chemotherapy we will not be doing that.
That's very good.
Thank you.